so thrilled to introduce very briefly Eddie Chang, as though Dr. Chang needs any introduction. He's a chair of neurosurgery here at UCSF. More than anything, he is such an innovator and directs the neuroengineering and prosthesis labs between Berkeley and UCSF, and he's going to talk us through any ideas about what neuromodulation technology could look like in the cancer space. So Eddie, I know your time is limited, so thank you for coming.
Sean, thanks so much for the invitation. And I think that this meeting is very interesting to me because you've really created this new intersection between two huge fields, one which I occupy in the systems physiology, human brain, and this really rapidly increasing knowledge about the molecular biology and cellular biology of gliomas. And it's exciting to see how these are interfacing in a way that I never predicted. So this title is really meant to be provocative, really think about some of the technology that we are using right now for physiology, both stimulation and recording, for the treatment of neurological diseases. And that's really where a lot of my work has been. And of course, none of this is really towards necessarily yet glioma treatments, but what are the technologies that may be relevant to the science that could be happening in several years? And also, what are the translational opportunities? So I'm not really going to get into the science of speech and physiology where we've spent the last decade really trying to understand and dissect, for example, the parts of the brain that allow us to speak or to understand words. We've made a lot of progress in the last decade, basically down to the point where we now know the electrical codes for every consonant and vowel, for example, in English. And several years ago, we decided to take a translational angle on some of our work to develop technology for people who are paralyzed and interface for people who have a paralysis of communication from brainstem stroke, ALS, or cerebral palsy. And in particular, a condition called locked-in syndrome, where people have intact cognition and awareness, but have paralysis of nearly all voluntary movement. And so this is a devastating condition, and a lot of us are thinking very hard about engineering solutions actually to bypass parts of the brain or the nervous system that are impaired to restore function. And so this is just a review that one of my former postdocs and I put together on why we are studying speech as a really unique way of restoring communication for people who have lost it due to paralysis. And the basic idea is that speech allows us to convey information at a much faster rate than any other modality by order of magnitude. And so by speaking, our species has allowed us to actually transmit information really, really quickly, faster than any other way. So our emphasis is really trying to restore speech, not handwriting or typing or that kind of thing. One of the first things that we did was a proof of principle in patients that were implanted with intracranial electrodes for monitoring and localization of seizures in our epilepsy monitoring unit. I primarily specialize in epilepsy surgery. And in many of these cases where we have to figure out where the seizures are coming from, we put electrode arrays on the brain called electrocorticography. And this is really the same technology that Sean has really taken off in terms of bringing towards the use of glioma electrophysiology. And one of the things that we basically asked in this one particular project was that if you could record high density electrocorticography from the brain, can you actually synthesize someone's voice from that brain activity? And this was one study where we basically showed that that's possible. The top is the spectrogram of what someone actually said into a microphone, just like I'm speaking to this one. And that's frequency as a function of time. And then below that is a second spectrogram that's based on a computer algorithm, largely an artificial intelligence algorithm, that is translating the brain activity into a computer simulation of the vocal tract. And then that computer simulation of vocal tract is generating words and sentences. So this is just an audio, like a movie demonstration. And if the audio works, you'll be able to hear how this sounds. If it doesn't, then I'll move on. Oops, let's see here. So that's the brain activity driving a virtual simulation of the vocal tract.
The proof that you are seeking is not available in rules. The proof that you are seeking is not
So the
available in
first was from
books.
the computer, and the second was from
The
what was
proof that
actually
you are
recorded.
seeking is not available in books. Shipbuilding within mas fascinating process. Shipbuilding is a most fascinating process. Shipbuilding within mas fascinating process.
So the point of this, and maybe relevance to this particular program, is that we're at this state where we can have electrodes directly on the brain surface, recording, and most of our work is in epilepsy tissue or normal cortical tissue, but also in the setting of brain tumors, and really providing perhaps a very special opportunity to understand how the brain tumors are interfacing with this normal function that the brain is using to create language, which is really fascinating. Because we had that proof of principle in hand in our epilepsy patients, we decided to start a clinical trial actually for someone who was paralyzed and had a stroke in the brainstem. He goes by the name Pancho, and the way that he normally communicates is by using uh, a stick that is attached to his baseball cap. And because he can move his neck, he can actually type things out word by word with what they call an assistive device. He can get out about 5 words per minute. And what happened was about 15 years ago, he had a brainstem stroke. The brainstem is the part of the brain that connects the cerebrum in the brain to the spinal cord and also to the vocal tract as well. And you can see that there's a lot of atrophy, shrinkage of the part of the brain called the brainstem, which allows all those signals and thoughts to be actually communicated. So when he woke up from this stroke after about a week of being um, in a coma, he could not move his arms and he couldn't talk. And he recalls that he wanted to die at the time because he was 20 years old at the time. So it's a really devastating condition, but he persevered and actually lives in a care facility where he can um, essentially communicate, like I just showed you, by moving his neck. So Pancho was the first participant in our Bravo trial. It's a trial that was designed to restore communication for people who are paralyzed using a permanently implanted electrode array on his brain surface. Everything that we had done was either in the operating room or in the epilepsy monitoring unit. This was really our first foray into permanently implanting essentially an interface into someone's brain. And this in particular case was a 128 channel ECOG array that was connected to a large refrigerator sized computer that would analyze the signals from Pancho's brain as he tried to speak words. So every time Pancho would try to think of a word, the computer would pick that up and then try to understand and decode which word he was actually trying to say. And so this is an example of the interface that he lives with now currently. Um, it's a percutaneous device, meaning that he has the array implanted on his brain surface, and then the wires are actually connected to a port that he's actually lived with for 3 years. So I smile because to neurosurgeons, this is like a nightmare in terms of thinking about, infections or other things. But he's actually lived with this for 3 years. And if you look closely at each one of those gold little dots, that corresponds to an electrode that's on his brain that is recording those signals from that very precise location. And then we can attach a digital head stage that translates those voltage signals into analog into digital and amplifies them and more. So the challenge that we have uh, that my lab spends a lot of time doing is translating the signals of the brain into things that we know as like words and text and that kind of stuff. And it turns out that we use a lot of the same technology that Google uses to translate from French to English. These models don't really care that much actually about French or English, but they're really good at taking a lot of data and mapping really complex relationships from one form to another. So the mapping that we're doing here is translating brain signals into words. And I'm going to show briefly a little bit how this works. So the top is a computer screen prompt where someone in the lab is going to write a question to him, and then the bottom is going to show you what is being decoded in real time from his brain activity. The blinking dots corresponds to detection. So the algorithm has detected that he's trying to say something, then it's trying to figure out what word he's trying to say. Okay, so we were really excited. This was the first time that this had been successfully demonstrated. There have been many, many years up to this where it had been attempted, but for various reasons was very complicated to do. And admittedly, this was just a starting point. So we started with a vocabulary of 50 words, and the algorithms use a lot of computer science. In fact, I would say even 5 years ago, the computer science algorithms didn't even exist in terms of artificial intelligence. So this is all very much in line with how technology is moving nowadays. One of the kind of technologies we use is called a language model. So basically, it's a statistical model of English. It's a probability of one word following the next. And so any of you who text, which is probably all of you, know that it will correct your text as you go. And the way it can do that is that it's got a model of what is the sequence of words supposed to look like. And so here, because we keep track of all probability of all words that he's trying to say, it can take that context and autocorrect the sentence like you just saw in this one particular sentence. The first couple of words were wrong partially. The top ranked word was wrong, but the second, third, and fourth were actually correct, and it uses that context to autocorrect and create the correct sentence. So that's what's called a language model. So all of that stuff is coming together. And one of the reasons I'm showing this is that, as we start thinking about technologies that will interface with patients who will have gliomas in the future, maybe we should be thinking about how to intelligently electrically stimulate in a way that goes way beyond the kind of treatments that they have for stimulation right now. Now, where is this technology going? Sean is very interested in getting to this, and our group was really the first, in addition to the Mass General group in Boston, to publish the use of these very advanced technologies called neuropixels that can record from single neurons, basically from hundreds of single neurons in the human brain. This is primarily designed for animals, and our lab and others have worked really hard to translate this into humans. So you can see that the electrode itself is tiny. It's about the thickness of a human hair, but it can record from thousands of sites. So this is a completely different kind of technology than we've ever used before. Traditional microelectrodes sample from one neuron or two neurons at a given time. This samples from hundreds. And why this is interesting is it allows us to actually record from different depths of the cortical lamina. And so on the left, you can see histology from brain tissue that we've removed. On the right are action potentials related to, and the waveforms related to single neurons that we've recorded in this technology. And each waveform has a specific profile that correlates to whether it's an excitatory or an inhibitory cell that we can categorize and just based on the signals. But some of the things that are pretty cool with this is that it's just seeing the single neurons fire, and each line here on this, what we call spike raster, corresponds to a single action potential that's fired by one neuron, just one neuron of the hundreds that are picked up by this thing. And I just want to play this video so you can see the kind of resolution that's recorded from this. This is a single sentence that's repeated over and over. And you're going to see this one neuron that really likes the sound N, the N and N.
It was nobody's fault. It was nobody's fault. It was nobody's fault.
We're kind of quiet there, and I'll show you another example. But we're really excited about this. This is not published work yet, but it's on the horizon. And having this kind of resolution to understand human cognition, I think, is really a game changer. So here's a different sentence.
He moistened his lips uneasily. He moistened his lips uneasily. He moistened his lips uneasily.
So that's exciting. And I don't know if those you could hear because it's kind of quiet, but there's this kind of noise and action potentials that we can hear. And if you look over those, 13 trials of that one sentence, the action potentials are really lined up to phonetic elements. So we're taking this neural coding question down to the level of single neurons and asking, how they code all the different consonants and vowels, again, at a newer level of resolution that we never had before. The last thing I'm going to talk about is other technology and projects very briefly that we've been using to treat neuropsychiatric conditions, which I think may be relevant to glioma care. in cognition, which is the use of implantable closed loop neuromodulation. And this was the first time this had been done, where we implanted a device into someone's skull and brain in order to record and stimulate to treat refractory depression. And the way this basically works is one of the electrodes is always sensing, or actually both electrodes are constantly sensing from the brain, and it detects when someone is having depression, and it sends an electrical stimulation, a small one, to help treat those symptoms. So this is what we call closed loop. And I'm part of a group with Collaboration Psychiatry, where this was the first time it's really done. This was a device that we used that's primarily being used to treat epilepsy, but now we're using it to treat depression. And the basic idea is the signals that are detected from the amygdala in this particular patient, which is really important for processing fear and anxiety, is used as a trigger to treat another part of the brain called the nucleus accumbens or the ventral striatum. And so when it picks up that she has these depressive or stress states, it will detect that and stimulate a different part of the brain. Now, this technology alone could be very relevant for the comorbidities of patients with glioma, but just trying to envision, as Sean has asked us to do, where is this going 5 to 10 years from now, where we can read from brain tumors their electrophysiologic signals, try to correlate that with patterns of growth or stability or quiescence, and then stimulate their neural activity in a way that can regulate whether they can grow faster or slower in a closed loop way, not just constantly applying stimulation, kind of like in the mindless way that we're doing right now with Optune and other technologies, right? So just thinking forward about how we can use neuromodulation tools now that are coming online, perceivably, if this whole glial tumor neuron brain thing is true, this is a natural extension of that we will be able to regulate tumor growth by directed electrical field modulation and stimulation. Of course, it's thrilling to be part of this kind of research. It's very difficult, of course, because of the psychiatric aspects of it. But these are a whole new generation of what we call brain pacemakers. So similar in concept to the things that are now very standard for heart, but now pacemakers for the brain and mind. And it's very intriguing to think about potentially pacemakers for brain cancer. So I just wanted to conclude there. This is just thoughts and speculations about how some of the work that we're doing just interfaces with the great group and team that Sean is putting together. Thank you very much.
Chris.
So I'm really excited to hear especially about the near transfer, far transfer. So near transfer closed loop modulation to help with these, conditions are very safe for our patients. How do we sort out whether we should start with near transfer or far transfer with rehabilitation or other sorts of, because we see efficacy with both, but we don't know who responds to what and what care pacing kind of cadence. How do we sort that out from where you sit?
When you say from a rehab perspective, yeah, it's an interesting question. I think the hardest part is trying to understand cognition. Some of these things that are proximal, like motor strength and even speech, I think are going to be in the near term. But cognition, I think, may end up requiring technology that is cellular, maybe, but we'll see. Yeah, but the technology is happening very quickly. The whole point is just to show where we are with some of these tools.
So given that, we know that neurons can actually activate brain tumors to grow, I think that's what Michelle Monti and so do you envision this technology therefore targeting specific neurons and inhibiting their activity directly to
I think
modulate
that,
growth?
I think that is one way to think about it for sure. And I think it's going to be more complicated, probably, where we're not trying to just inhibit activity, but allow normal activity, but trying to downregulate the kind of pathological activity that might be driving some of this synaptic growth. But I think that's going to take some time. You got to figure out what those electrographic signatures actually do. There's also this interface with seizures and epilepsy. That interface with epilepsy and too much.
That's sort of the low-grade glioma, IDH, alpha-ketoglutarate, 2-hydroxyglutarate, metabolites.
It's all starting. to make
It's all
sense.
starting to make sense.
There are preclinical models for frequency of stimulation for tumor growth or whatever, but nothing obviously in the clinical space,
Right.
which is why this is super, super exciting. Andy, you had a comment?
Yeah, I just had a thought about this. Great presentation, by the way. But just thinking about the inherent difficulties of using a system like this to eventually slow down proliferation. What about the thoughts with using something like this for directed growth of tumors, where it would be us like trying to gear the tumor to regions of the brain outside of the eloquent cortex or places that are more easily resectable?
Yeah, I mean, I think that that's a really fascinating idea of how to control the plasticity of the tumor and its interactions with the brain. And that will be 2.0 for sure. But to just imagine that.
Thank you. Thanks, Eddie.